All right, everybody's joining on now. So good morning, welcome to VSPG. Today we have a presentation by Dr. Jim Schiffbauer.
Alex, did you start recording?
Yes.
Okay, good.
Yeah. So Jim, you can go ahead and start sharing your screen.
Sure.
Whoops. So today from Dr. Jim Schiffbauer on tubes and taphonomy from the terminal Ediacaran of Southwest North America. We had a cancellation last week, or for next week, but we now have Professor Aki Johannesson is going to present next week. And let's see how quickly I can pull up the title just to let you all know. And his title for next week is things you can do from home during a pandemic. A geochronological review of magmatism along the external margin of Columbia and in the Grenville age origins from the core of Rodinia. So going for that next week. But today we'll go ahead and I'm going to introduce Jim for us, or unless Andre had any other announcements, but I think we have it covered. So I'll go ahead and introduce Jim. Dr. Jim Schiffbauer is a paleontologist primarily interested in animal evolution and taphonomy across the Ediacaran Cambrian transition. He uses his expertise to critically analyze the fossil record with close attention to the fine line between true biological information versus preservational artifact. Beyond traditional paleontological techniques, Jim includes geochemical methods and analytical microscopy methods such as SEM and tomographic X-ray microscopy. Jim received his PhD from Virginia Tech and working with Professor Shuhai Zhao on Ediacaran tubes and embryo fossils from the Gaojiashan and Doshantou formations in South China. After his PhD, he continued at Virginia Tech for his postdoctoral research, and then started a position as research assistant professor at the Virginia Tech Nanoscale Characterization and Fabrication Laboratory, and he remains involved at Virginia Tech as an adjunct professor. But in 2012, Jim started his current position as assistant professor at the University of Missouri in the Department of Geological Sciences, and has additionally served as the director of the University of Missouri X-ray Microanalysis Core Facility since 2017. It's really great to have you, Jim, another Virginia Tech alum here, like Andre and I. So you guys get to hear from another Virginia Tech Hokie. So
Proud
please
Hokies.
Go ahead, Jim.
Yeah, absolutely. Thank you to everybody for coming in this morning. This is great. I have to admit, usually I am teaching my historical geology lab at this time. So I've been keeping up with everything on YouTube as soon as they're posted. And I got to come in last week, of course, for Fred's when we were on spring break. So, but today I'm going to talk to you a little bit about some of the work that my group and I have been doing on tubes and taphonomy. And I'm going to start, if I can get my slides to move here. Everybody see everything okay? Does that look all right? Good? Okay, good deal. I'm going to start out with just the acknowledgements, of course, as we tend to do. We work in large teams. A lot of the effort that you're going to see today is collaborative with many people that I've worked with over the past decade. And this is just a small list. My group on the left, and then, of course, my external collaborators, Yao Ping, Ben Yang, Emmy Smith, Lyle Nelson. Some of you are in the room today, so great. Thank you. I appreciate you coming. So hopefully you'll see some new things that we're doing, as well as some of the old things. And I'll start off just kind of simply with an introduction to what I do in my lab and the efforts that are continuing with all of these projects. I guess kind of I'm most known as a taphonomist. Most of my research, I think, falls into that general header. And it's not just Ediacaran and Cambrian, although those are where a lot of my biological questions lie. So I tend to be focused in those areas, but I have been expanding into the Paleozoic quite a bit more recently, and getting into, I don't know when exactly this happened, but I've become a taxonomist as well. I'm getting into arthropod taxonomy. I've been doing a lot of tube taxonomy, and I think these are all directly related to the preservational questions that I ask as well. So it's all kind of combined in together. And then, of course, as Alex mentioned in the intro, I am the director of the X-ray microanalysis lab here at Mizzou. And this is just an example of some of the really cool things that come through our doors. We get to work on a lot of amazing samples. So like I said, I'm expanding into the Paleozoic. I guess I'm expanding all the way into the Cenozoic as well. This is some work that I'm doing with some colleagues across campus, describing a hominid species from about 3 million years ago. And what you see here is a 3-year-old boy's jaw of this hominid species. And you can see once that video plays again here, you'll be able to see the fade. So you can see the adult teeth forming inside of that jaw. And hopefully this is forthcoming soon as well. So this will be a fun paper to get published. So let's get into the main thrust of what we're here to talk about today. And the big overarching problem that a lot of my group has been working on is the appearance of these skeletal tubular fossils in the terminal part of the Ediacaran. And we're trying to make sense of the diversity of these organisms. We're trying to make sense of what they represent and how they may be related, and thinking about this on a global scale. And I have to say thanks to Fred for last week. He gave a great intro on, or an overview, I shouldn't say just an intro, on how these things are correlated globally. And this is kind of the direction that I think a lot of this work needs to go, is thinking about what exactly these organisms are and how we can make sense of their appearance all over the planet at the same time in the last part of the Ediacaran. And that kind of untangling that relationship between the organisms of the Ediacaran to the organisms of the Cambrian at the Cambrian explosion is something that I think a lot of us at some point in our careers have had questions about. It's one of the biggest outstanding questions in the history of life itself. So I think this is a really fun and exciting time frame to work on. And we'll get into more of that. This is, I unapologetically screen grabbed this from Fred last week. This is a great figure, showing a lot of the diversity of these tubular organisms that we'll talk about, and highlighted here in the apricot that you can see on the left. That's the Ediacaran organisms, and then those in the springtime green on the right are, of course, those that we see more in the Cambrian. But those lines, I think, are being blurred as we're getting more organisms. We're finding more organisms of this time frame and figuring out what they represent. And there have been a lot of areas of growth recently in this work on these terminal Ediacaran tubes, from thinking about their taphonomy. This is some work that I did with Ben Yang and other colleagues, summarizing a lot of the preservational modes that we see, as well as the morphological characterizations that kind of unite this group of organisms. This was from our description of Zunia from Mongolia, which is known from the lower part of the Cambrian. And we have been kind of moving further into phylogenetic approaches as well. This is not from my group, this is from Taeyoon Park's work, discussing the potential relatedness or phylogenetic positioning of these cloudinids and cloudinid type forms with the octocorallians potentially that we see in the lower part of the Cambrian. And I think these efforts are worthwhile. We need to, the more things that we can start to compare these with, the better understanding ultimately we're going to have of this picture. We additionally have seen Frankie Dunn's work not long ago in Nature when they were describing Aurora Lumina. This was a really cool paper that included some discussion on some of these tubular forms at the end of the Ediacaran, and indeed included a morphometric approach as well. And you can see that here and highlighted in the light blue are the Ediacaran forms, highlighted in the orange are the Cambrian forms. And this is an attempt to try to figure out where these things are plotting in three-dimensional morphospace. This is using an NMDS analysis to try to resolve some of the relatedness of these morphological features. And ultimately, I think this is it highlights a really kind of important issue for these organisms, and that issue is on relatedness versus convergence. We tend to, in the literature, talk about these things a lot as cloudinids, which suggests that there is some phylogenetic relationship between them. That's using the Hahn and Flug family name effectively to unite these organisms. And so that idea echoes that they are in some way, shape, or form related to one another through evolutionary history, and that might not exactly be the case. If we do just a quick survey of the literature, we can find that tube forms are rather convergent across a number of different phyla, and I'll show you examples of that momentarily. But this led us to introduce a term which is ultimately a semantic issue, of course, the cloudinomorphs. And the idea here was to try to separate convergent morphologies from hints of phylogenetic relationship or implications of phylogenetic relationship. And we followed suit with Mark Laflamme and Shu Hai with regard to how we offered the name, for example, comparative to the Ernietomorphs or the Dickinsoniomorphs, et cetera. We wanted to try to keep it in line with what had been done in the Ediacaran to this point. And the goal here was to simply say, look, there are a lot of organisms that share this general construction of funnels and funnels, or tubes and tubes, or barrels and barrels, or whatever we want to call them, some type of collared unit sitting inside of another collared unit, nested to grow an elongated tube. There are a lot of these things, and to assume that they're all phylogenetically related is probably a step too far. So I think that grouping them as a morphotype is probably more appropriate, especially at this time. And this, I think it follows off of one of the lines that I co-opted from Frankie's paper, placing Cloudina in the total group of any animal phylum may be premature, and I agree. But I think the same should probably be said for all of the cloudinomorphs more broadly, and even more than that, all of the tubular forms in the Ediacaran. This is a lovely example of Phoenicia here from the Ediacaran of Australia. And these are weird. We don't know what they're related to, and trying to connect all of these dots has been difficult. So like I said, if we do just a cursory search of the literature, you're going to find that tubiculous organisms, tubiculous life modes are relatively common, and this is across numerous phyla. We have showing examples here of phoronids on the left, and most of these are modern with the exception of that one Oesia or Margaretia doris, as a lot of us still recall it being named. But we have phoronids that live in tubes today. We have anthozoans that live in tubes. Obviously, Oesia is an enterobnust that has been interpreted to live in the Margaretia doris tube, which had formerly been an algae. We have sibiglinids that live in tubes, and these guys look very much like a lot of the cloudinids that we see with their funnel and funnel constructions. So there are a lot of possibilities of things that have acquired this tube-dwelling life mode that I think we want to be cognizant of when we're trying to offer biological interpretations of what these organisms may or may not be and may be related to. So the big question, I think, is, at this point is, what do we do from here? How do we start to resolve this biology? Can taphonomy tell us anything? And I think these are all big questions that we've been trying to approach several groups, and I think we're starting to offer a lot of insight on these types of things. This is just a simple character matrix table that Selly and I put together in some of our early descriptive work a couple of years ago on the materials from Nevada that I'll highlight through most of the rest of the talk. And this was the point of this table was to talk about some of these morphological features that these tubes have or don't have, and how they could potentially be compared. This was simply for reference for people that were continuing this type of work, and where we could, what generic level inference we can make depending on what features tubes that you find may or may not have. And what we've been doing since that, and this has taken several years, and we're still not ready to publish it, but we're working on it. We've been putting together a massive database. I'm thankful that I have a colleague in the department here, John Huntley, who I went to school with. He was a Virginia Tech Hokie as well. And he's kind of known as a database guy. He puts together large databases of, he's done this with acritarchs, he's done it with predator-prey interactions, et cetera. And this kind of came from simple discussions that we were having over lunch one day about the potential to do a morphometric database on the tubes, the terminal Ediacaran organisms. And so we started to build this. This has been a big effort from all of the students that I highlighted on the first slide in my group, as well as others, Morris and Nolan from Shuhai's group, and Xing Tong, and several other folks that have helped with this, Natalia Baikova. And we've been data mining. That's exactly what we're doing. We have, at this point, over 150 references and 117 different taxa that we've described, and we're describing every single bit of information that we can from these, from basic morphological details to preservational or taphonomic details to lithologic details, how they're preserved, where they're preserved, and all of the metadata that goes along with this. And so far we're closing in on 20,000 cells of data. And we're hopefully going to have something soon to show everybody. Our initial exploratory statistics look something like this. This is NMDS again, similar to what Frankie reported in their paper. Frankie's data set, by the way, is it's a presence absence data set that was largely derived from the table that Tara and I put together in that JSP paper. So this is kind of a build from that. We're trying to add loads more data to get better at to see this picture better ultimately. And kind of to walk you through this, what we're seeing from left to right on NMDS1, on axis 1, is a separation by form. We're getting organisms that I would call tubomorphs on the far left and highlighted here in blue, which include things like Cynotubulites and Gaojiashania. These are generally tube and tube type construction organisms. And I will also again say, Fred, that your figure was the gift that keeps on giving. I stole all of these little highlights, or these little organisms that I've included here from that figure as well. So as we move from left to right, those tubomorphs grade into the conomorphs, which are generally more conical. These are things like Cambritubulus, largely. I've used Anaborites here as the example organism, but of course that's a bit different of an organism than most of what we're talking about in this group. In the pink to the right, or the salmon color, those are the broader cloudinomorphs, and then highlighted in that darker color or darker shade of the same color are what would be called Cloudina proper within the genus Cloudina. And these are the organisms within the cloudinomorphs include a lot of other things like Saarina and Phainella and Multiconotubus and Conotubus, et cetera. Lots of different things there that are constructed generally in the same form. NMDS is largely driven by size, and there's not a whole lot of separation, as you can see there. So, but this is a summative figure of all of the information that we have. And what's largely driving that axis 1 is the general construction of the tube. And hopefully we're going to explore these, we're going to dive into this data set a little bit more with different exploration techniques to figure out what we can resolve. And I think another important bit of this is to not just do it broad scale with all of the tubes, but dive into some of the individual groups as well. For example, I think Cloudina itself needs some more exploration. It occupied a relatively broad range in that figure that you just saw. But if you take a look just across the species that have been defined within the genus, this is work from Xu Chai not long ago, I think 2021, where this was published in a survey of the cloudinids. If you just kind of draw your eye to the scale bars here in this figure, what you can see is, for example, Remkeae has a 50 micron scale, whereas Lucianoi and Carinata have millimeter scales. And these are large, 10 millimeters for Lucianoi, 5 millimeters for Carinata. So these are up to 2 orders of magnitude difference in scale that we're talking about with these tubes. So to try to unite these under the same genus, I think is rather difficult to do. And I think we need to really kind of dive into how we've been classifying these to this point. And hopefully that NMDS will help draw some of these things out once we've had a summer to sit and explore as we hope to do this summer. And I think this work is ultimately important if I want to kind of highlight the big picture of importance at this point, kind of in the somewhere in the middle of the talk here, is that it's not just for understanding biology as it pertains to the Cambrian explosion. It's also, this is going to be a useful tool because all of the proposed subdivisions of the Ediacaran stage, the terminal Ediacaran stage, excuse me, have been pinned on the appearance of these skeletal metazoans in the fossil record. So I think understanding their correlation globally is going to be of utmost importance for us to, as the ICS and the Ediacaran subcommission, to truly offer a formal subdivision of the Ediacaran period. So let's get into what they are. This is where a lot of my work really lies at these days. And the big thing is, to this point, everything that we know about the cloudinids, about the cloudinomorphs more broadly, has come from their tubes because soft tissues are not found pretty typically. So we make a lot of inferences based on what the construction of these tubes. But as you recall, I just showed several examples of tubes across different phyla. So this is problematic. And depending on how we, the amount of weight that we place on certain characters that we define is going to change our inferences as to what these things actually are. And this is something that people have been doing for quite some time. This is a really great example from Vin and Zaton's work back in 2012, so just a little over a decade ago. And this was relatively simple, but it was important nonetheless to look at some of the characters that Cloudina proper has and try to relate those to similar characters that we know in modern tube-dwelling cnidarians and modern tube-dwelling calcareous polychaetes. And ultimately, this was just a presence-absence data sheet that said that it probably fit better, Cloudina probably fit better with the cnidarians than it did the polychaetes, on the basis of some of the microstructural details like granular microstructure and the closed terminus of the base and things of that sort. But I think that some of the more recent work, Luke Perry, given this is only a couple of years after the Vin and Zaton paper, this was 2014 by Luke Perry, highlighted the evolution of the annelids quite well in both what we know and, as shown here in the solid lines, those are the known fossil record of these groups, and then the presumed molecular divergence times in the dashed lines. And the organisms that Vin and Zaton were largely comparing these cloudinids to fall within the sabellids or the serpulids or the ceraculids. And we don't have a fossil record of those until we get into the Carboniferous. So there's a 200 million year gap that we need to explain, I think, between the Ediacaran and the origin of those groups specifically. So I think perhaps we should be looking a bit further in the record, or other things that, other annelids potentially that would be more appropriate comparators for the cloudinids proper. Now, if we go into a lot of the features that have been published or resolved since the Vinen-Zeton work in 2012, we've got a lot of features. A lot of new things have been reported. This is a great example of one from Amy Shore and Rachel Wood's work on multiple tube branching in some of these cloudinomorphs. And this was from serial grinding of sections of material from Namibia, and what they were able to see is that sometimes we have individual tubes that branch more than once. And we've known about cloudinid branching for a long time. That's not something that's new, but something that is new is that they could branch multiple times. So this was argued very, very nicely in Amy's paper that this could be suggestive of a hydrozoan affinity, because we don't know of any annelids that branch multiple times. If we're just, again, we're just thinking about the difference between cnidarians and annelids at this point. But as soon as we find things like this, the biological world answers back, and they offer new roadblocks in these types of interpretations. And this is a great example of that. This is Ramacilius. It was published shortly after Amy's paper. This is a multiple branching annelid. It's not a tube dwelling annelid, but it is an annelid nonetheless that branches multiple times, and each of those individual segments has a terminus or an anus, and those individual segments can break off, and they're usually the reproductive unit of the organism as well. So it's an interesting organism, and it's unlike many other annelids that we know. Of course, this is the first reference of multiple branching in annelids. So the biological world tends to have answers for as many of the characters as we tend to throw at it, which is rather interesting. We've seen dichotomous branching. Like I said, this is something that we've known about for a long time. Ivan Cortillo, who you probably know from the reports of Cloudina carinata from Spain, put together a really nice paper on the differences in dichotomous branching that we see with regard to two basal structures that we seem to have apparent in the same deposits, where you have a rounded terminus in some cases, which he suggested was likely a result of sexual reproduction, and then a conical terminus, which he suggested was from asexual reproduction, so from the dichotomous branch or the budding of the organism. So this is what was nice about, what I think is a really nice contribution about this paper, is it talked a bit about dispersal mechanisms and reproductive strategies as well of the cloudinids. So this is something that when we look at their global distribution in the terminal Ediacaran, we need mechanisms to explain how these organisms got all over the planet simultaneously. And this is kind of a good way to show that, I think. In really extremely well-preserved examples, this comes again from Ben Yang's work when we described Zunia. We were looking very closely at the ultrastructure of the tubes, and we found that the tube walls in many of these cases, especially in Zunia, had nice organic layering, fine laminar structures within the tube walls themselves. So we expanded that search outside of Zunia, and we looked at Cloudina from Cloudina that Ben had from Namibia as well as from Paraguay. And we found the same thing in these exceptionally well-preserved examples, that there are occasions where we have organic laminae within the tubes themselves, within the tube wall, which is a little bit different than the granular microstructure or the calcification that we commonly see in many of the other examples, where I think taphonomy plays an important role in how those appear. We can find the cloudinids and in these dense aggregates. They've been interpreted by Rachel's group. Amelia Penny specifically led this paper as these potentially being reef structures, or a biological construct here, where much like worm reefs today or other coral reefs, where we have large abundances of these organisms in the shells growing together effectively. Akshay Mehra and Adam Malouf suggested alternatively, though, looking at the same materials and through serial grinding, that these are probably not biological constructs, and instead detrital buildups. And this is something that we see in a lot of cloudinid deposits today, there are a lot of these coquina type deposits where we have just tons of shell hash kind of thrown together with reckless abandon from a storm event or something that has transported them to their resting place. And it wouldn't, it's obviously not a biological construct at that point. But I think that the jury's still out on those kind of things. These are the types of questions that we need to keep asking and keep assimilating data on. And this is an older one, but it's still an important one. We see potential predatory drill holes in the cloudinids. These lovely round circles that Brian Pratt and Hua Hong had shown in some of the Chinese materials. They're exceptionally well-preserved, nice rounded structures that we presume are from drilling organisms, drilling predators, and it signposts a really important increase in trophic complexity, and it foreshadows, of course, the evolutionary arms race that we see in the Cambrian shortly thereafter. So I think owing to a lot of these different novelties, this is why people tend to say that these organisms heralded the Cambrian explosion or laid the groundwork or were key players on the stage of the Cambrian explosion. However you want to phrase it, I think they're all valid ways to think about these organisms. And so what we've been doing is, of course, this is a drill down into a couple of specific localities rather than thinking about a global scale. But I think these kind of efforts are things that we need to keep doing around the world where we have really nicely well-preserved organisms. And what we've done in Nevada is we've been taking a look at some old fossils. Figuratively and literally, this is the work that Taylor and Phil Signor and Mark McMenamin and Hagedorn and various other folks have done to describe all of these back in the 60s through the 80s. So we're talking, 50 years ago in some cases. And we've been trying to bring these back to the forefront a bit. And I think that comes a lot from Emmy Smith's work back in the late 20-teens, 2016 and 2017, when she and Lyle had reported a lot of really cool, slightly different fossils. These were preserved a bit differently than the stuff that Taylor and Phil Signor had been looking at years and years ago. These are all pyrotized, and there were similarities drawn between the Gaojiashan population and the community that we see in Nevada. And so somewhere along the line, when Emmy was publishing the 2016 paper, the editors at Geology asked me to write a research focus, given my work in the Gaojiashan. So this really kind of started a communication between Emmy's group and my group, and trying to figure out what we can do to describe these things as best we can and classify them. So this is what we focused on, and we found from the initial work that at the time one of my students, now she's faculty, but Tara Selly, worked on. It was we were looking at the two most abundant forms that we had from Emmy's collections, and on the left you can see, hopefully these are the kind of the standard funnel and funnel shapes that we would see, that we would call cloudinomorphs anywhere else. And on the right we have a slightly different organism that seems to be more barrel shaped. It's less tapered. It's interesting. It was initially grouped with Gaojiashania in Emmy's original description, but when we started to look at this, it's constructed quite a bit differently. So we dove into these organisms and we offered a couple of taxonomic placements for them. We named the one on the left after James Hagedorn from his work in the region. Tsarina hagedorni, and also from Emmy's undergraduate work with James, and he served as a mentor for me as well during my PhD. He was always a reference for asking questions and figuring out how to use technologies like CT and to answer questions that I might have. And then the one on the right, owing to its general appearance as a stack of tires, we named it after the Michelin Man. And here's your fun fact for today. The Michelin Man is actually the oldest corporate mascot in existence. It was first used in 1894, and he actually has a name. It's not the Michelin Man. His name is Bibendum. So we named that Costatubus bibendi after the Michelin Man. And the other fun fact, I guess, that's associated with that is bibendi actually means to get drunk. That's where it comes from. That's the slogan was, now is the time to drink. I don't know why that was the slogan for a tire company, but it is what it is. We can I digress. When we were going through the taxonomic description of a lot of these organisms, we were trying to gain as much morphological information as we had, or as we could get. We were using micro CT to do it because we wanted to see, these were three-dimensionally preserved for the most part. We really wanted to see how these things were constructed and offer a better three-dimensional characterization as best we can. And what we found was something really super exciting. And in the lower part of the screen, you can see that. This is going through projections of the organism, and we found that there was a very distinct tube inside of the external skeletal tube. And knowing the literature, looking at preservation of guts and other soft tissues, this kind of screamed to me that it was potentially soft tissue that we were looking at inside of this tube, and we really wanted to figure out what it was. So I'll walk you through this figure. What we're doing here is this movie that we're building out the rock through the stacked projections. This is the external tube, and that inner part is the internal tube. We're dissolving away that rock, and we're going to relief. We're just going to have here the segmented view of the exterior tube in red and the interior tube in gold. We're going to clean up all that extraneous pyrite that's in the rock itself that has the same general grayscale. And there you go. That's what we see. This is the captured view. And I have to say, I have to admit, this is really, honestly, the very first one that we scanned had this structure in it. So it was really exciting, and we scanned a whole bunch of them afterwards because I needed to figure out what we were looking at. And ultimately we inferred that these were guts. And if they are indeed guts, if we were right with that inference, it kind of does point us towards a direction in biological interpretation, or what we can say about them. It would be a one-way through gut, much like we see in polychaetes or annelids, and even in Pterobnus as well. But many of the other expected tube dwellers to have existed at the time had either U-shaped guts or had two-way gastrovascular cavities like anthozoans, which are structurally quite a bit different than what we would see, or what we would expect to see for a one-way gut, which is what we suggested. But the taphonomist in me, it was kind of a compulsion. I needed to figure out how these things were preserved. And as any good museum's nightmare would do, I promptly cut this sample up because I needed to see it. I knew I wouldn't sleep if I didn't. So I took the best one we had at the time, and I sectioned it so I could really look at how this potential soft tissue was preserved, and I could drill down on it as best I could. And that's what I did, and that's what I'll show you here. These are the individual sections of that tube itself. The exterior part is the skeletal tube. The interior part is what I've got highlighted in the black boxes here are the internal soft tissues. As I zoom in on those, hopefully what you're going to be able to see, and I'll zoom in a little bit further. First of all, they're pyritized. This is pretty clear in backscatter imaging. You can see framboidal structures on many of these. But secondly, what you can see is that there seem to be both centripetal and centrifugal growth of the pyrite from a nucleation point, which I suggested was the gut wall itself, which maybe was histologically more suitable for pyrite growth and nucleation than other parts of the organism. But for whatever reason, it nucleated on that. And hopefully you can see, as highlighted in between the two black arrowheads, that there's your tube wall or the gut wall. And then the white arrowheads are pointing in the direction of pyrite growth away from that, both internally and externally into the cavity of the organism itself. And this, kind of started making us ask questions about the overall preservational quality at this locality and what we could say from the taphonomic perspective. And so we started to investigate a lot of these. We needed to collect more. We needed to survey what these what the taphonomic expression really looked like across the entirety of the unit. So this is what we've been doing. And we, from my past work of the progression of pyritization, I started to think about, because about 90% of the fossils that we find from this site are pyritized, I started to think about how pyritization proceeds and how it's limited, and how we may be able to back out some information from that with regard to understanding how these guts might have been preserved, if they are indeed guts. And pyritization can be limited by three different things. It can be limited by organic matter, if this is driven by bacterial sulfate reducers. It can be limited by iron, of course. If you don't have reduced iron in the setting, you're not going to make pyrite. So you need to be in an anoxic setting to begin with. And it can be sulfate limiting. Again, if it's bacterially sulfate, if it's driven by bacterial sulfate reducers, you need sulfate to fuel that BSR process. And thinking about how the taphonomic expressions would follow depending on which part of this sequence is limited, we can have a few different outcomes. If organic matter is the only limiter, then pyritization can effectively proceed until all of the organic matter is gone. You're not going to stop pyrite growth simply because you ran out of iron, for example. It's going to continue until there's no more organic substrate for the bacterial sulfate reducers to grow on. In the other cases where you have one of the building blocks of pyrite that's limiting, then you can cease kind of partway through the process. If iron is limited, you're not going to stop bacterial sulfate reduction, but you will stop the production of pyrite. If sulfate is limited, you are going to stop bacterial sulfate reduction at some point. And I think that those are the cases where you're going to end up with partially pyritized materials, which is really what we have in our case. If you take a look at the Gaojiashan materials as an alternative, most of them, like this cross-section that you see in the bottom left, are completely pervasively infilled with pyrite. So there's not really a chance to preserve any of those soft tissues. If they were preserved, they would have been overprinted by the process of continued pyrite growth. Whereas in our case, in Nevada, what we're dealing with is really relatively limited pyritization, but it's usually limited such that it's capturing the three-dimensional morphology of these organisms, but then kind of stopping in a lot of cases. So this has been a kind of what we call a Goldilocks scenario for capturing these types of features. So where we don't have to worry about either too much decay or overgrowth of mineralization, this is kind of the just right scenario, where we're going to get the best preserved or the highest likelihood of preserving those internal soft tissues. So to kind of walk through that process quickly, because I'm already running out of time and I need to get through some more slides. So effectively the process, the model that we are proposing for how these are preserved is pretty straightforward. We have the initial burial of the organism that is, and what we're looking at here, let me kind of walk you through this before I get too far into it. This is the organism, and we're going to keep going through the organism view as it's buried. And this is the cross section of the organism where we have the outer tube, and then we have the organism itself, soft tissues in the pink and that digestive tract in the red. So what we think is happening during the initial burial event, and we think this was a relatively rapid burial event, is it's going to reorient the tube to be prone to the bedding surface, because we don't believe that these were rigidly biomineralized. We think there was some flexibility in their skeletons, and we see that during compaction. I'll show you that in a moment. But if we bury these fast enough, we're going to get past the potential rapid degradation from oxygenic or oxic living microbes that are pretty efficient at decaying carbonaceous materials. So if we get it buried to the bacterial sulfate reduction zone, I think we're in pretty good shape, because that's a little bit less efficient, but it also is what makes the pyrite production work pretty well. And in this case, what we're dealing with is sulfate reduction. Sulfate reducing microbes are going to start to chew up all of that organic material. You're going to start to lose some structural integrity of the organism itself from the decay of all of its soft tissues. But that initial pyritization is going to occur on the most suitable localities, histologically suitable localities for pyrite growth. And we think those are, for whatever reason, for better or for worse, they are the gut wall itself and the tube walls. We see this pretty well in the Gaojiashan, that the tube walls are indeed a good locus for pyrite growth, but we just don't see anything inside, but we do here. We see that continued pyritization, like I showed earlier, kind of growing centripetally and centrifugally from the gut wall, and that's effectively what we're showing again here. In this portion, as that continues to grow, we get a structure that is relatively robust, that's going to not get compressed too much or destroyed. And we actually see that the compression affects both the outer wall and the gut wall approximately the same. So we think this happened prior to sediment compaction because they were compressed at approximately the same rate and same shape. And then finally, we have cessation when sulfate reduction runs out, or sulfate runs out. And we also think that the positioning of these guts as they fall towards the bottom of the tube are probably a geopedal indicator, such that they're going to gravitationally settle once the soft tissues have kind of decayed away, that we're supporting the gut in the center of the organism, or presumably in the center of the organism. So that's kind of the overall view of the taphonomy of how these things go. But I will say, as we've been working on the taxonomy of these organisms, taphonomy is proving to be a really complicating factor in understanding the diversity that we actually have. And I'll give you the best example I have of that. This, I think all of us would agree, looks just like the Saarina that we named after James Hagedorn. This is, we have stacked funnels. We can see the funnel rims really well. It's pyritized. If we throw an elemental map over top of it, you can see that rather well. You can see funnel flares, even like we do in most of the cases of Saarina. Now, we micro-CT'd this sample, and I'm going to show you that data next. And I kind of slowed it down, and I'm walking through some projections here, because the morphology is going to change as we go from the top, the upper part of the bedding surface, to within the bedding surface, the lower portion of this organism. So here you go. This is longitudinal view. Looking at this, you can see all of those funnels rather well, and we're moving downward into this organism. We're about halfway down now, and all of those funnels have disappeared. And now this looks like a smooth tubed organism, something that we could probably classify, if we didn't have any other information, as something like Cambrotubulus, if it were the same size, with a smooth interior and exterior lumen. And that's tough. That's a change in morphology halfway through the organism, probably related to taphonomy, and potentially related to the same process that I just talked about, where we have soft tissues are settling towards the bottom. In this case, I think we had more sulfate reduction and probably mineral overgrowth on the bottom side of this organism that kind of eradicated those funnels. I don't think that they are, I don't think the funnels themselves are decayed away. I think the pyrite overprinted them, so we don't see them anymore. And this is one of the things that we're working on writing up now with some of these other forms that we're finding, and hopefully trying to classify as well. And we've seen a lot of different things. The more we look into these after or outside of the Saarinids and the Costatubus, we're seeing a lot of other forms that are worthwhile of describing, as rare as they might be. We have examples that look a lot like Multiconitubus, where we have fully nested funnels that you can see in the bottom. That's an example of Multiconitubus from the Death Valley or the Nevada localities. Whereas at the top is what we had originally described from South China on that genus. We've seen other organisms that we are grouping with Baculophyca and Rugatatheca, which are not stacked funnels, but rather just wrinkled tubes or straight elongated tubes, some of which have a holdfast like Baculophyca. I'm doing my absolute best. The reason that there's a Mars Attacks logo or still here is, I'm not sure this is your other fun fact for the day. The Martians originally landed in Pahrump, Nevada in Tim Burton's, what I would think is his magnum opus, Mars Attacks. So we are working on naming these species after characters from Mars Attacks, just to give Tim Burton some more credit in the phylogeny or the taxonomic paleontological literature. We've also seen maybe even Phoenicia, and this is a really interesting thing. Our samples on the right, we have CT. Once that rock spins back around, you'll be able to see it. And then the one up towards the top of the screen is, of course, from Australia. One of the things that I really need to do, though, is I need to get an Australian sample for comparison to see if it's structurally the same. I'm looking at the list of folks that are in the room today. Hopefully Rachel can send me one of those, or Mary. But I think that the external morphology does look really, really similar. And but the problem is we only have a couple of them. We've only found 2 or 3 at this point, and that's exceedingly rare. But this interesting internal tube is something that I'm not quite sure about if it exists in the Australian examples of this as well. And then finally, we're expanding, and this is again to keep co-opting Fred. He talked about the La Cienega formation a bit in his talk last week, and the recent paper from Hodgin et al. With Lyle and various others on that. There are comparable organisms that were found in Sonora, Mexico, that McMenamin back in the 80s, again, compared to what we know from Nevada and California, and reported most of these as cynotubulids at the time, although they were reassessed by Grant and then again by Ben Yang to suggest that they are probably not cynotubulids. So we've been kind of diving into these a bit more recently with materials that I got from Sarah Pruce, and we're able to take a look at the morphologies of these from extracted fossils. I will say this one hopefully is forthcoming pretty soon. There are some really interesting things here. Number one is that all of the extracted materials that we get are silicified, and they're very coarsely silicified. So it's hard to make heads or tails of a lot of the exterior morphologies that we see because of how chunky they are. But there are dolomitized examples as well within the same rock. It's just that when we dissolve the rock, we're not left with the dolomitized ones. Those go away with the rest of the coquina. So we're doing the best we can with these, but I think realistically, we're looking at several different taxa here. We have what I'm pretty confident I could agree with Mark and say that these are cynotubulites. We certainly have also Cloudina that Grant suggested. I think we might have Conotubus there as well. If you look at frame 4 in this figure, I believe the kind of imbricated funnel rims there are different from Cloudina, and they're of a different scale. These are larger fossils than the Cloudinid fragments themselves. And I think that looks a lot like Conotubus from the Gaojiashan, which is curious. We have some smooth tubes there too that we really can't say much about. But we also have some other interesting features. A lot of these are compressed or torted. And we also have what we think might be punctures in some of them, much like the drill holes that had been reported from China as well. So with that, I'm going to go ahead and move to a take-home message and wrap this up. And I don't think I can make a take-home message in words that's any better or more appropriate than this figure that Rachel Wood had put together in their Nature, Ecology, and Evolution paper. This is a phenomenal figure that really kind of highlights that both of these organic walled tubes and the biomineralizing tubes at the terminal Ediacaran occupy such an important temporal range in between the soft-bodied classic Ediacara biota and then what we know in the lower part of the Cambrian and towards the Cambrian explosion. And they occupy so many important evolutionary novelties, from potentially the first guts that we see in the fossil record to the first animal grade biomineralization to maybe predation to maybe reef building to a lot of different things. And regardless of where you stand on any of those debates, I think we can all agree that they are incredibly novel, incredibly evolutionarily important, and understanding their diversity and position on the tree of life, I think, is something that's quite important to do. So with that, I will say thanks again for the invitation, and I'm happy to take any questions you guys might have.
Great, thanks a bunch, Jim. It was good to have you, and it was a great talk. And we will go ahead and we'll switch to the question and discussions. So you can ask a couple ways by either typing your message, and I can ask it for you, or you can raise your hand with the button at the bottom. If you find reactions, click the up arrow, or sorry, click on reactions, and you'll see raise hand at the bottom there. We do have several questions, hands up, but I also had some stuff sent to me during the talk. So I'm going to start with one from really quickly from Karthik Sender Sarwan. He sent 6, but I'll just start with one for now. And maybe
I'll do my best to answer all 6.
I'm also just gonna, he sent them directly to me. I'm going to send them to you because there are some big words.
Sure.
Maybe you can read them off aloud before you answer them.
Let's see. Wow. Those are big words. You're not wrong. And I don't have a big word answer for that question. So there's structure of paramorphicline structure or petamorphicline structures indicating subsequent decrease or increase in calcium concentrations in proximity. I think those are these are obviously with regard to the evolution of biomineralization. I think that there are a lot of questions that need to be addressed, and I think that yes, I think there would be some good indicators of calcium. Yeah. I don't have a good answer for you. I'm not gonna try to make something up because I don't want to off the top of my head get bogged down in discussion of calcium concentration. But yeah, I think that it's a fair point, because we see differences, of course, in the relative biomineralization of these types of organisms from locality to locality.
Okay, Karthik, you had a lot more to say, but I'm gonna swing through to get some other people as well. So go ahead, Prescott, you have your hand raised. Please unmute yourself.
Hey, Jim, thanks for the talk. I had kind of, I guess, a 2-part question. The first part is about the guts that you're finding preserved in these, and it seems like they're relatively 3-dimensionally preserved. And I was wondering if you could speak at all to the contents of those guts, if you can determine anything, if they're sediment filled or fluid filled, and what that might be able to tell us about how these things are feeding. And then the second part is kind of more just a general curiosity question, is how does an organism with a through gut reckon with having a biomineralized shell that is closed at the apical end?
Those are both great questions, Prescott, and good to see you. So to start with the first one, yes and no. So I think I saw Rudy was in here earlier. I'm sure Rudy could talk about gut preservation and content better than I can. But what I will say is that what's curious about these guts is that we see them in a couple of different ways. We see them hollow, or relatively hollow, I should say, where you've got some sediment infill, but nothing out of the ordinary. There aren't any bits of anything that I'm seeing in there that would be identifiable. So there's not like, oh, well, it ate some bit of something else that we can recognize. There are other cases where the guts are, we only have, so let me give you the numerical. We only have 6 guts total, but we've only CT'd of the saurinids that we have these in. We've only CT'd about 60 of them. We've kind of shifted to CTing other things, so it's about 10% where we're finding them. But it's pretty clear when we find one in the field, like we'll actually write on the rock, CT this one because it's probably got something, or it's a good candidate to have something. So sometimes we see them completely infilled with pyrite within the gut itself. And that one that I sectioned is kind of curious because it has both pyrite infilling and sediment infilling. So I think that it's possible to start talking about the life mode of these things as potentially being filter feeders or things that we know today that are tube-dwelling polychaetes similarly that do feed from suspension in the water column. A lot of those are also deposit feeders when the suspension runs out, when there's nothing that where they're hungry, but there's nothing to eat in the water column. They'll kind of flop their palps over outside of their tube and feed on the sediment, the surface of the sediment, hoping that there's organics there. So I think that might be a mechanism to get some of the sediment that we're seeing inside of the gut. We don't see big blocky cementation or anything like that most of the time. So it's unique. Anyway, now the second part of that question is, how do things like this live in closed base tubes and have a one-way gut? That's a good question, and it's something that's been pointed to a lot. A lot of folks have asked that question. Surprisingly, a lot of polychaetes today have a closed base, even though they're discussed often as having an open base. And I will tell you why. If you have an open back end where you're advertising that you're an organism by shoving off waste out of a hole in the back, a lot of predators will find that, and they'll find that they can get in that way and take you out. And that's not a great strategy for continuing to survive. A lot of these organisms, sabellids and several others, serpulids, they have ciliated fecal grooves on the underside of the worm itself. And when they expel waste in the base of the tube, when it gets to be too much, they move it out of the front of the tube, or the aperture of the tube, the top end, by moving that through their ciliated fecal groove to the top of the tube. And also, these things can move. So like I said, they can come out of their tubes and feed on the sediment and organic material that might be on the sediment around the tubes. They can also turn around in their tubes and eject waste out of the top. They're not anchored.
Cool, thank you.
Yep.
Okay, great. Rachel, your hand was up next. Please go ahead and unmute yourself.
Great, thanks, Alex. Hi, Jim. It's good to see you. Great talk. So I have like a two-part comment and a question. In terms of the Phoenicia, as we've talked about, it's interesting. Phoenicia does have a hollow interior, which we know from the presence of smooth-walled internal molds and collapse, but there's some other features in your specimens that I think are a little disparate, but we can talk about that later.
I agree.
Yeah. So coming to my question, I think, as you obviously agree, the broader morphogroup of tubular organisms should be divided into smaller submorphogroups, right? But I think there's still the question of how we're defining a tubular organism in that broadest of morphogroups. So I just kind of was wondering how you would define it and what the motivation behind including things like Nomicolathus is within that broader group.
Honestly, Nomicolathus, we included it because it's weird and different. So it was kind of to have an outlier. So if that's a fair answer to say, it had nothing to do with anything else. It was simply, this is another skeletonizing organism in the late Ediacaran that is different and demonstrably different. So let's include it and make sure it doesn't fall within these other groups. So trying to define these broadly, this is, it's been something that is hard. We've been, like I said, we've looked through, we're over 150 papers. I think we're at almost 180 now. And we're just trying to take everything that everybody describes kind of within this temporal range, within this construction range of generally tubular. You know, there are a lot of weird organisms at the time, like Shanksalites, which when we reported it initially from the Gaojiashan, we talked about it being a stack of discs, right? Which is totally different than a tubular organism. But now there are some other reports of Shanksalites that look a hell of a lot more like cloudina, and they've been suggested to be potentially related to. So I think there's a lot of taphomorphic overprint on a lot of these things too, which is why we're trying to, it's why I'm trying to do these things at multiple scales, to look at big, broad scale and at fine, let's look at this one specific locality scale, to try to get a better resolve on these. And I certainly think that we're including multiple different types of organisms within these, right? Baculophyca, for example, is maybe an algae and not a metazoan. So we're dealing with a lot of those kinds of disparate, you know, groupings and trying to make sense of them. And that's very difficult to do. But hopefully we'll chip away at it. I don't think I have all the answers, and I don't think I'm going to have all the answers. I'm hoping that I make a contribution that people can use to further ask questions.
Right. Yeah. I've been struggling with the same thing. So I was hoping to just get some insight from you. So thank you.
Yeah.
All right, Greg Ritalik, you had your hand up. Go ahead.
Yeah, great show, Jim. The shift show
Thanks, Greg.
prevails. Love it.
Good to see you.
Yes, I was glad to hear Rachel thought that was not Finisia, because I agree. Which
Yeah.
brings me to the question. Have you ever seen any of these tubular things in non-pyritic red beds, hematite-rich red beds?
I wouldn't say so. Most of what we've been looking at are siltstones to finer grain mudstones. I don't think any of them are you know, the iron concentration in the rock when I'm doing simple spectroscopy stuff is relatively low compared to some other rocks that I've looked at in the past that I would put more along the line of hematitic. So I'm not seeing anything necessarily like that. With regard to the Phoenicia specimen, I agree. I don't know what it is. The closest I can come up with is it kind of looks like Phoenicia, and I think this was I think Lyle might have Lyle's in the room, so he might have better knowledge on it than I do. This was from him and Emmy had collected this, and then we found two more on the last trip that I took. But when I CT'd the other two that we found, it doesn't look anything like that. So that was the one-off that is very strange, and I can't put my finger on exactly what it is. But we're trying.
And it's a Mount Dunphy specimen, right?
No, that's Montgomery Mountains is where that was from.
Okay.
Yeah,
Okay.
yeah. So closer to Pahrump, over in Nye County, not Esmeralda.
Yeah. No, I've been searching my mind, and I don't know of any of these tubular fossils in non-pyritic red beds either. So if there's one that you find, let me know. I'd like to explore the exceptions.
Yeah, yeah.
But thanks for the talk. Great job.
Yeah, no problem. Thanks for coming.
Okay, so we don't have more hands right now. But I have several things in the chat that I can bring up. Let's get one of these other ones from case underscore 1, and then LaRossi Abril.
Rudy
just put their hand up, but let me go ahead and give you another from case underscore 1. This one says, I'm putting it in the chat as well, but I'll read it. Would mutualistic interactions between true cloudinids and other taxon, similar to the biotic fouling on ventulicolians, help us in classifying or grouping the cloudinomorphs, i.e., by assuming the mutualistic interactions are biologically specific to a taxon?
Yeah, so I think that's a good point. there, and I think this is, so one of the things that we are doing in the broader database project is trying to think about how these communities are structured, what the community makeup looks like in each case. In a lot of cases, they're relatively, I don't want to say monotaxic, but they're relatively low numbers of taxa within these groups or within these communities, and others are very diverse, even if the diversity some of the diversity is very rare comparatively. I will say the cloudinomorphs tend to be dominant in most of the localities I've looked at, where those are generally the most abundant organism compared to others. But we are also looking at, this is one of the things we did, or I did with Yaoping Kai when I was a student. We looked at microbial mat related lifestyles of some of these cloudinids. We published that, I think, in Precambrian research, maybe. So I think that there are a lot of potential possibilities to look at how these things were living, where these things were living, and with what taxa they were living with, to try to think about, affinities a little bit more, with comparison to modern organisms and their mutualistic, commensalistic interactions.
Okay, great. Rudy, go ahead, please.
Hi, I hope you can hear me.
Yep.
Yeah, awesome. Yeah, sorry, it's kind of improvised. So it was awesome. I really loved it. It's a very good summary about all these very intriguing fossils. I wanted to ask you two quick questions.
Sure.
The first one is that, do you still believe that some of them, or at least a good part of them, had a biomineralized tube? Because I've seen this comment in, I think it's in Young and others in 2020,
yeah,
where you kind of like give the feeling that you start doubting about this, like maybe it would have been almost purely organic tubes, like tough tubes, like chitinous or this sort of thing. So what are your thoughts about this these days?
Yeah, yes. I'll straddle the fence on this one. So I think that there are taxa that are demonstrably mineralizing, but there are, of course, taxa that are not, or are lightly or less biomineralizing than others. We see this quite a bit in the Gaojiashan. We see, reasonably well mineralized cloudina towards the Beiwan at the top. But then, conotubus and things of the sort down in the middle member they're organic. They're robust. I don't know if I would say necessarily chitinous, but they're recalcitrant for sure. There are plenty of modern examples, sabellids and others, that are definitely organic walled tubes. And we're starting to see some of these cloudinids with really, really nicely preserved walls have organic layers as well. So I think, thinking about how they're structured, how the tube is being built, I think it makes sense that some of them might have had an organic component to that, or a template that mineral grew from, whether or not they were, super rigid or not. I think, we're seeing a lot of pliability or a lot of plastic deformation rather than, brittle deformation.
Yeah, I agree with you. I've seen that on some of your pictures, for instance, and
yeah.
I was thinking the same, like if it's a biomineralization, at least in this particular fossils, was probably a light biomineralization. We got the same thing sometimes with arthropods too, like a light biomineralization and this kind of reaction to deformation. My second question is that, and I'm sorry if it's like a very naive question. As you know, I usually work like on younger beds, right?
Yeah, the other side of the Cambrian boundary.
Yeah. Well, I'm still very interested in what you do in Nevada, right? Like in the, some of these things can be found also in the basal Cambrian, which is very interesting. Are they found in special depositional settings? I mean, especially in terms of bathymetry, do they correspond to like maybe organism living in particular environment that
Yeah, so that's a really good question, and it's something that we've been trying to parse out as much as we can in Nevada, in the Dunphy area, and in Montgomery Mountains, because those are two different bathymetries or two different water depths from site to site. In Dunphy, most of the fossils, most of the pyritized fossils, I should say, are coming out of shalier units, so a bit deeper. And then in the Montgomery, they're coming out of siltier units, so probably shallower. But I think realistically, and again, not to continue to use the Gaojiashan as a correlative, but it makes a good one in this case. You know, we discussed the biostratinommy, how these things are being transported and buried. So I think that we're not capturing them necessarily in the place that they were alive.
I understand.
I think we're getting storm events that are wiping them out, ripping them up. And, you know, and you can see this not just in the way they're preserved in sometimes relatively dense aggregates, but you can also see it in the fragmentation of the tubes, right? So a lot of times we're missing the back end of the tube or the apical end of the tube because it seems to be unrooted or broken off from wherever it was attached originally. Yeah, so it's tough. But it's, you know, we're kind of our search image has been more about where we expect to find pyritization, at least my teams, because that's what we're, you know, kind of we're investigating the taphonomy along with all of this. So that's kind of where we've been focusing.
Yeah. Awesome. Thanks a lot. Thanks a lot.
Yeah. Good to see you, Rudy.
Yeah. Good to see you too. Yeah.
All right. I can go ahead and give us another from Karthik. How do cloudinomorphs compare to the encrusting Rhenalsis forms around the base of archaeocyathid sponges?
Oh, you know, honestly, that's an organism that I've not looked at enough, especially considering that Rowland's Reef is right there in Dunphy, and I've not I've not done enough on the archaeocyaths at all. So that would be something that would be really curious to go look at. I know one of my colleagues here at in my department, Sarah Jacquet, she's been working a little bit on the archaeocyaths, but mostly in the small shellies that are preserved around them rather than anything within the reef. So kind of laterally extensive from the reef instead. So I can't speak to that, but I should take a trip and get some archaeocyaths.
All right, let's hit the next one then. Is the growth in cloudinids similar to ecdysis? If yes, would instances with more cups or older individuals have an inherent preservation bias associated with them due to the increased area for pyrite growth?
So I think that there's, I still think that the progression of growth in cloudinids is something that we don't have a great grasp on. I would say that most of the time we tend to think about these things as maybe adding another funnel at the end if they're getting longer. You know, if we use modern sabellids as an example, you know, they kind of reach a point where they stop growing more tube. So I don't really know, but I will say more towards the preservational part of this. I think the bigger the organism gets, you know, the more focused bacterial sulfate reduction you would need to preserve it. The more volume of pyrite you would need. Of course, you could do back of the envelope calculations on how long that would take and et cetera. So I think it'd be tough to start getting preservation of some of the bigger ones. But I think also that it's again relates back to Rudy's question about being dislodged and repositioned somewhere else during, you know, rip up and burial during an abrution event or something, some kind of storm.
All right. Then there's a couple more. Let's go ahead. Can the coelemic arrangement of cloudinomorphs be understood by inferring the pyrite nucleation sites?
Can you say that again? I'm sorry.
Can the coelemic arrangement of cloudinomorphs be understood by inferring the pyrite nucleation sites?
I think so. It's what we, that was at least part of my model, right? So trying to figure out how the coelom looks, how it's structured within the organism is kind of all wrapped up in the thought process of how these would proceed through decay and lose structural integrity, et cetera, towards getting to that pyritized flat-lying tube at the end against the skeletal wall. So I think these, I would hope that we could find some more information that if there were coelomic divisions or anything of that sort, that, if those wall boundaries were something of the sort could also be preserved through pyritization, that would be something we would see. But I don't know. I don't know if we can truly resolve it, but I make some assumptions based on what we've seen.
Okay. One more with relation to pyritization. Do we see any pathologies associated with cloudinids, especially something similar to pachyostosis? If yes, how does the pyritization change in these cases?
I don't have a good answer for that. So I tend to try to dissociate pyritization from what the organism is necessarily. I tend to try to think about it in a purely microbial product zone, if that makes any sense. So, I think there are potentially histological controls, but I think those histological controls are true for every single mineralization process that might replicate soft tissues or fossil organisms. You get into kinetics of mineral growth in those kind of conversations. What kind of differentiation in or the pre-existing organic templates would favor, I don't know, silica growth in silicified organisms or calcium carbonate growth in calcified organisms, et cetera. I think those are all comparable kinds of questions, and I don't necessarily think that it matters as long as the process to make those minerals is occurring, if that makes sense. I don't know if that answered the question or not.
Seems that I think Karthik would put more in the chat if we had anything. Seems, but great, cool. Prescott has another hand up. Go ahead, Prescott.
Yeah, absolutely.
Hey, Jim, I just thought of another question. I was curious, so some of the modern analogs that we use or relate to cloudinomorphs have both biomineralizing and agglutinating forms.
Yeah,
and I was wondering, are there known agglutinating cloudinomorphs from the Ediacaran, or do we think we'd even be able to confidently recognize them in the fossil record or expect to find them there?
So that's my question, is how do we confidently recognize an agglutinator in the record? Who was I working with? Lucy Muir and Brendan McGavin. We described an agglutinator from, I can't even remember, sorry. Anufianella was the name of it. I cannot remember the locality, but it was not Ediacaran. And it was a tough one. It was a tough sell because it was an agglutinator. So it was, a lot of folks in the review process asked about whether or not we were really looking at a tube or just some bits and bobs of other materials outside of the standard depositional material there. So it'd be tough. It'd be tough to recognize. But I agree with you. Yes, there are certainly well-known agglutinators on the planet today that build tubes comparably by just sticking sediments on to their exterior.
Cool, thanks.
Yep.
All right. So we're
Um,
currently out of hands and out of questions. Anybody else?
Jim, maybe I ask a question. little bit out of my expertise, but I understand that all of them you consider to be attached, or do you think some of them could have been floating? And if and either way, how do you think they spread from one continent to another?
Yeah. So those are really good questions. Even with regard to whether or not the tube was upright or lying flat on the sediment and recumbent, those are big questions. Bruno Becker-Kerber and various others, they describe Cloudina as flat lying, as recumbent on the sediment with only the aperture turned upwards. But I've commonly referred to it as the alternative way. Shuhai used to talk about this as a cobra form, where it's kind of flat lying for one part and then it curves upward, and you have that kind of the top of it that can feed in the water column. And I think it could be really dependent on the organism, right? As to what it, which life mode it actually occupied. If they are building reefs, pretty commonly we would expect those reefs to be somewhat upright, or the skeletal tubes to be somewhat upright within those reefs. And they would be kind of growing upward into the water column and tearing to ultimately get more access to different nutrients and whatever else, and competition-driven dynamics there. I don't think any were floating. I don't think any were pelagic. I think they were all on the sediment in some way, shape, or form, or even within the sediment. There are some examples like Gaojiashan and a few others that we kind of tend to see in cross-section. They have a flatter bottom than top, so we think that they were probably laying on the ground, and that flat bottom corresponds to the sediment-water interface where they were laying down. With regard to dispersal, I think we have to think about trying to get a juvenile or larval form from one continent to another is going to take a lot of water current to move it. So that's pretty tough. But at the same time, we have some analogs that we can take a look at in the fossil record, and in the not too distantly removed fossil record from where these things were living, like some of the interrupnusts like Herpetogaster and various others in the Cambrian that we think are sessile, but we find them in the Burgess, we find them in the Chenjiang, we find them quite far apart from one another, even the same species. So there were ways for larvae to disperse that far, and I think that it's trying to figure out, what those larvae looked like is the question, right? And if we have them preserved.
Thank you.
Yeah.
Okay, Jim, you've been really popular. I've got another one sent to me in the chat, but I'm going to call that the very last one so that we can get you a break here.
I don't have anything on my calendar until 12 o'clock, so I'm fine.
Okay.
I like talking about tubes.
Yeah. I can definitely tell. So case under Sarwan asks again, do we find stratification, niche partitioning reflecting on the tube in cloudinid individuals of the metazoan reef of the Nama group?
So is Fred still in the room? That might be a question for Fred. I've not looked at the Namibian reef materials as much as I probably should have. I have a whole bunch of materials here in my lab from John Grotzinger's collection. So that's something that we are hoping to do. But I would say that, Fred and Amelia and Rachel would probably know better at this time than I do. Hey, Fred, I see you now.
Jim, can you repeat that question? I just missed the first part of the question.
Yeah.
I do my best.
I'm going to real quick. There's the question and then there's a follow-up to it.
Thanks so much for the amazing talk, Jim. I really enjoyed it.
Hey, I mean, I stole a bunch of your figures, so
You're welcome.
So he says first, do we find stratification and niche partitioning reflecting on the tube and cloudinid individuals of the Metazoan reefs of the Namib Reef?
So I can't speak directly to that either. I wish Rachel was here to answer that question, but I would say that the reef, the one possible reef that we've looked at in the Nama comes from one specific site called Riedornflachte. And although there are loads of individuals that are all over the place on that reef, there's absolutely huge outcrop. The one feature of the reef that wasn't described so much, following on from that 2014 paper, is a crypt was filled with early marine cements, and it's a void space, and it's quite a big one as well. So that was one of the key features that we used to infer the possibility for reef building. I would also say that that entire section, it's only about 30% exposed on the surface. It's tilted and it's buried. So a lot of the material that you're seeing there is actually reworked material. There's no doubt about it. So whether or not this is a real reef building area is still, I think, slightly up for debate. So, but as far as niche partitioning goes, I think that Rachel would have to answer that. I think I really can't speak so much about that.
Thanks, Fred.
Okay, cool. Well, since we've run up to 9:30, let's go ahead and call it there. Thanks a bunch, Jim, and thanks a bunch for all the really good discussion.
Yeah, thank you all. That was great. That was fun.
Thanks.
All right, and see you all in weeks to come. Take care.